Welcome back. General Ecology. If you're if you haven't caught on to that yet, this is chapter 22, Biodiversity, and it is the last chapter and the last sort of series of lectures for the course this year. And we've been going down a list of topics that are highlighted in the text, and I've been kind of digging into them a little bit from my perspective. We were talking about the equilibrium theory of island biogeography, how we got to that theory and how it helps us with preserve design. And now we're moving into the topic down here, global scale biodiversity patterns. And a little bit more about what drives biodiversity. We've already talked about the idea that area, proximity to a source population, are important, but there's a little bit more going on here as well. So what is it, for instance, about area that, other than the fact that you can cram more things on a bigger area, or maybe it's not cramming, they can diffuse out and eke out their existence without being crammed up. More than that, there's something else going on. Okay, so let's look at that. We're going to start with the patterns of diversity in North America, and you can see that the total number of species is highest, you know this too, in the tropics, and it declines as you move towards the poles. So whether you're talking about mammals, reptiles, trees, amphibians, mushrooms, as you start near the tropics, I guess these are near the tropics, the equator's down here, but you get a pretty high level of diversity, and then it falls off as you move north, right? You know that. Okay. Oh, what the heck, I'll share this with you. So if you tie a bunch of strings around the planet, this is one of the theories as to why that pattern holds. Okay? So you tie a bunch of strings around the planet on a globe, that you have in your house, and they're all along from the poles down to the other pole, north to south. Okay? Now you start, and you put a little weight on the end of each string. Okay? And now you start spinning the thing. You'll discover that those strings wind up through centripetal force, winding up along the equator. So some would argue it's because of the spin of the planet. Those are probably the flat earthers. It's the spinning of the planet that forces the species down along the equator through centripetal force. And they further that theory with the idea that the reason you have such large mammals in the north, those humongous polar bears and other things, versus all the little tiny mammals that you see in the equator, is because of centripetal force, the polar bears drag and they don't make it all the way down to the equator. Again, these are the flat earth people, but there you go. I just thought I'd throw that out there. No, it's not any of that going on. What's really going on here? Okay, we've already highlighted this. The richness is seen higher for animals and plants in the south, or along the equator, and it goes down. Richness goes down as you move to the poles. So this is written from the other perspective. It's increasing from north to south. That is, from the North Pole down to the equator. And let's see, the other feature here, and you can see that in this panel A, is that for mammals and reptiles and things, you actually get increases as you move east to west. There's more diversity. Not so much for amphibians, not as much for reptiles, but mammals, trees. Well, trees are pretty high here. It's really the mammals. Why? Well, look at the pattern of where the high diversity is. That is the darkened zones in mammals on the west side. And if you know anything about your geography, you know that this is the Sierra Nevada in California, and this is the Rocky Mountains through a variety of different states and even up into Canada. That's probably driving it, okay? And that's because of increased habitat diversity, heterogeneity when you get to the mountain ranges. And remember, these mountains are getting up to 14,000 feet, going from high alpine treeless zones all the way down to the deserts and prairies and things like that. So there's a tremendous variety of habitats, which then allows for a tremendous variety of mammals to hone in on those different habitats. Important feature. This, of course, is occurring in the oceans as well. We've seen examples of this figure in other lectures. If you compile all the records of marine organisms around the world, you can see that the highest diversity is in the tropics again, kind of in this whole zone. And look at this. Man, if you want to get into biodiversity studies, especially in the oceans, but it's also on all these islands because there's a lot of islands, a lot of habitat diversity, etc. But in the oceans, this region is super rich for a variety of reasons. And that Song of the Dodo book also highlights the fact that there's a trench here and there's not a lot of interplay between the diversity of Australia and the diversity here. Yes, a little bit, but not much. There seems to be a break there for dispersal. Okay, and the lowest diversity is in like Central America. Well, no, that's high diversity. Less diversity as you move south and north. Although, look at over here along China. Diversity is still pretty high along the Ring of Fire. Okay, the underlying pattern is being driven by heterogeneity, right? Habitat variation. These two examples give this stark relationship. You have so much water, so much nutrients, so much light in this marsh, but it's almost depauperate in species richness. There aren't a lot of species there. Why? Because the habitat is pretty boring. It's all super rich. And only a few species are there are a few species that are super competitive and can push everything out that decides to try to get established in that beautifully rich habitat. These guys outcompete them, and it's a low diversity site. Go out to the desert. This is the Sonoran Desert with the saguaro, and there's so much diversity through the ridges and the amount of water and soil types and all kinds of different things going on there, rocky versus soil, etc. You get far more diversity. Now, of course, they're showing you these two pictures. I guess they're both at blooming time. Maybe this is the marsh grass that's blooming, but obviously this is grass. These aren't grasses. There are all sorts of different shrubs. There's a blue one there, yellow, probably several different yellow ones. That seems to be a good color for pollination. So they're kind of pushing this image a little bit, but it's true that the deserts, I've already said, if you think deserts are boring, go out and spend a few nights and days. Stay cool, stay dehydrated, stay hydrated, don't get dehydrated. You will find that it's a beautiful place to be. Very diverse. Birds, mammals, reptiles. Just have to know when to look, not at the heat of the day. Okay, right. Okay, so just kind of hits one more idea on what drives diversity. Habitat heterogeneity is huge, but PET, potential evapotranspiration, tells you how much energy is in the system. So this is giving you an idea of how much plant activity, if you will, evapotranspiration, both evaporation and transpiration. And what's going on is it's a function of the moisture in that habitat. So you're going from 0 to 2,000 millimeters per year of potential evapotranspiration. Again, you reach a plateau. Diversity is really increasing, that is species richness, as you increase the PET, and then you reach a plateau and it's like, well, there's a lot of variation, but you're not really increasing all that much anymore. You've kind of maxed out, the plants have. Okay? But with those plants comes additional structure. There are researchers that have looked at, just from the base to the top of a tropical tree, how many insects you have, and they partition by height on that tree. So you can imagine that there's a lot of habitat diversity if you've got a lot of PET, potential evapotranspiration. Okay. This kind of gives you the same idea, but it does list the different kinds of organisms to show you that it's not just the plants. You still have PET as your measure on the x-axis, potential evapotranspiration, but now you're looking at richness for birds in red, mammals in dark blue, reptiles in light blue, amphibians in green. So they're all going up at different rates because they're different kinds of organisms, from low evapotranspiration to high, and they all plateau at some point. Note the reptiles kind of take the longest to get there. Birds plateau out pretty quickly. Okay. So, another thing that I like to highlight with respect to diversity, and I've already kind of hit one issue that I have with how we measure diversity. Species richness to me is a rather boring measure. Just counting up the number of species doesn't really do it for me because it doesn't get at other aspects of that diversity. In other words, abundance. I've already hit home on that, and that's why we like the Shannon-Weiner or the Simpson's diversity indices, and there are others that give us abundances, not just the number of beetles, which you can count these up. I think they're probably not all beetles. Insects, a lot of them are beetles, but I don't know about this guy down here. Entomologists are probably cringing. But anyway, it's not just the number of insects, but how abundant are they? So I don't see a mosquito in there, but it can be really abundant in some locations. And that makes an impact on our livelihood and our willingness to stick around, right? So abundance is important when you talk about diversity. How do we measure those species is also really important. This is the other big thing that I like to highlight, and I think I have highlighted. I hope you aren't tired of this storyline, but the morphological species concepts aren't really helpful in a lot of cases. So maybe that's not the best approach. What do we do when we're doing the morphological species concept and we go down to the tropics and try to find a new species? We collect it, we preserve it, we put it into an herbarium. That doesn't do a lot in terms of understanding what that thing does. If it's a very rare species, okay, that's all we had. So we find Sasquatch, we collect her, we put her into a museum and call it good. Would have been much better to wait around and try to figure out what are the functional roles that species is providing. And so it's not just collecting of the numbers of the species for our diversity, but maybe we should look at functional diversity and not even worry about the species. That's another approach that a lot of people are taking. What are the functional things going on in this ecosystem? What are the different functions? And then get at who's doing them. That's super powerful, super hard, super time consuming. That's an entire career, which is a fantastic career. Super helpful. We could also kind of tied in with that get at the metabolic diversity. So remember my tree of life? I'll have one coming up shortly. The metabolic activity. What are the different organisms doing in this ecosystem? It kind of gets at the functional role. Those are kind of nicely tied up. And then the thing that I've honed in on my research is phylogenetic, because the morphology isn't doing it for me. And what I can't get morphology usually. I mean, at least insects have a couple legs and different colors and, you know, some things going on with their morphology that we can, and their sexual parts are really important. But I don't have that with fungi. In fact, we usually don't have the mushrooms around. So we pull up a little bit of hyphae and we say, yeah, this is white versus black versus brown, not doing it. So we have to extract the DNA and then put it into a phylogenetic framework and then say, oh, there's a lot going on here, or no, these things are all the same or similar. So phylogenetic is very helpful, and it can feed back to functional because closely related species probably have similar functional and metabolic activities. So that helps. Okay. So then that brings us to the phylogenetic diversity, and I showed this in the first days of the lab, or of the lecture. And And I do want you to remember that we at ESF are super happy to stay right up here, but the functional roles are pretty limited amongst us, the plants, the animals, the fungi. Yeah, there's some pretty big breaks. The fungi are more similar to each other than the animals are to the fungi, and they are to the animals, and the plants have their own branches over here. But within each one, they're pretty similar. Plants photosynthesize. Sorry, botanists. I know I'm simplifying this down to something that's probably offensive a little bit. Animals and fungi basically take up nitrogen and store it for a while. Take up some carbon that the plants photosynthesize and store it for a while, then decompose. The fungi are decomposing and helping plants and being symbionts and pathogens and all that. But you get the point. It's not quite you've seen them all, but there's a limited amount of information that you might pick out of these. The phylogenetic diversity tells you that. Versus all these other eukaryotes, and the branch lengths are meaningful. These things are bizarrely different than anything we have, whatever this is, right? In the basal proteus, what's going on there? We don't even know. We can't even, most people are not all that interested. They're little tiny microorganisms, but they are probably doing some pretty amazing things for us. And we have stories that are coming out that that's in fact true. The archaea, right? This group of bacteria are as different from bacteria as they are from eukaryotes, or either one of them are from eukaryotes. And then the functional roles here, also look at the branch lengths. There is some serious stuff going on there. And if we want to protect the planet, we ought to be focusing in on that level of diversity and try to get at some new hypotheses to test. So stop with that soapbox, and I'm going to come back next time for the final bunch of slides looking at a little bit about biodiversity and how to protect it, or how we resource what we have to protect these things and what's the best strategy. I'm not going to solve the problem. It's a small part of this lecture, but I want to throw some ideas out to you guys for you to think about as you move forward to solve the problems. Thank you very much. Okay, cheers. We'll see you next time.